Oh.
All right. Welcome, everyone, to the library. It's my pleasure to introduce our speaker tonight. For those of you that don't know, I'm Matt Dunn. I work here in the library, and I thank you all very much for coming. I'm thrilled to be introducing Ann Skalka. She is a very familiar face here at Cold Spring Harbor. She started off, she did her undergraduate work at Adelphi doing field biology with salamanders. But after graduating, she went to start off her graduate work at Yale. Then her advisor fled across the country shortly thereafter. So she started work at NYU with Jerry Hurwitz. And after he moved to Einstein Medical College, she finished her PhD and graduated from NYU and started her postdoc here under Al Hershey. And she was here with Alf until his retirement about 5 years later as part of the phage group. And I've always wondered, when you become a member of the phage group, did they give you like a hooding ceremony with a viral capsid? That's the kind of thing that keeps me up at night. Then, after leaving Cold Spring Harbor, she took a position at the newly formed Roche Institute of Molecular Biology in New Jersey. While here, she was studying lambda phage and actually was gave the first demonstration of the temporal and spatial control of transcription, which is amazing. And after staying with Lambda for a while at Roche, she then switched to Retroviruses, which we're all very excited to hear about. And she stayed there for about 20 years before moving on to Fox Chase, where she stayed for about another 20 years. And
30 years.
30 years. My apologies. That's a marvelous career, and I can't wait to hear all about the book. So everyone, please, let's give a warm welcome to Ann.
Okay. Put my water down. Well, I thank you, hardy souls, for coming to listen to this little talk and for your interest in my book. The book was written primarily for students and for the science-interested public as an introduction to a fascinating field. And in thinking about a theme for this book, I was struck by the realization that simply by asking questions about the unique properties of these viruses, we've gained just an enormous insight, not only about their biology and their ability to cause disease, but also the very origins of life on Earth and the factors that drive its evolution. So my goal was to share these insights in a way that would be accessible to both experts and non-experts. And my strategy was to first prepare a basic understanding of the science involved, and that's the first couple of chapters, and then include cameos, little stories about the major figures and how they actually made their discoveries and some of the struggles they faced during and after they made those discoveries. So the book has, to tell you a little bit about it, has 7 chapters. And the first couple of chapters are a kind of an general introduction, and I thought I'd focus on those for this evening. The first chapter tells the story of the early pioneers, and in a period when they discovered these things called viruses, and they had no idea even at that time what genes are encoded there, and whether they're encoded even in nucleic acids. So retroviruses were first discovered in 1904, and that was really only 6 years after the discovery of the first virus, which was TMV. And these were very mysterious agents. People knew about bacteria then, but these things weren't bacteria because they would pass through a filter that would retain bacteria. And they also wouldn't grow in broth. So they were really quite strange. But the first ones that were discovered were what we now know as EIAV, which is actually a lentivirus that's related to AIDS virus. And it was discovered by two French vets who were treating animals who had something called swamp fever. And it was a kind of anemia in horses. And they discovered that they could pass the infection from one horse to another by using these filtrates. And then a couple of years later, about 4 years later, a second virus was discovered in chickens. And again, it was a leukemia, so it was a blood disease by two Danish scientists. And they also discovered that it was this filterable agent. Now, neither of these things caught the attention of the medical community then because, well, anemias weren't considered to be cancer at the time. And who cared about horses and chickens? I mean, it didn't have anything to do with human beings. In 1911, a little bit later, the third virus, called the Rous sarcoma virus, was found by Peyton Rous. And this virus did catch their attention because it was obviously a virus that caused a tumor. And even though it was chickens, it really did make an impression. So a little bit about Peyton Rous. He grew up in Baltimore and got his medical degree at Johns Hopkins, right there in his hometown. And but after he got his medical degree and did his internship, he decided that he didn't really like clinical medicine. He didn't really like dealing with people. And so he took an assistantship as a pathologist in Michigan because he wanted to work in the laboratory and not bother with people. And then after about 3 years, he then went to Europe, and then he was invited by Simon Fletcher to come to the Rockefeller Institute, I think it was called then, and now it's Rockefeller University, because Fletcher was interested in starting up a unit that focused on cancer. And his friends told him that was a complete mistake because that's a dead-end area of interest. But he was a young guy, and he was only 4 years after his medical degree, and so he took up the challenge. And I think that most people who study retroviruses know the story about the discovery of Rous sarcoma virus. Shortly after he arrived in the Rockefeller, a woman came who was a farmer, a nearby farmer, and she had a hen that had a huge tumor in its breast. And he looked at it, and being a pathologist, he realized that this was a sarcoma. And he thought, well, he'd heard about these filterable agents causing disease, so he decided to test that. And what he did was take out the tumor, chop it into little bits, and then implant the little bits into other chickens of the same breed. And he discovered that these chickens also got tumors. And then he ground up the tissue and made a filtrate, and he also found that this filtrate would produce tumors in the chickens of the same species. And so he thought, well, this really has to be cancer. And he actually had two publications describing a cancer in chickens that was caused by a virus. Well, the findings were very, very controversial. First of all, well, maybe he had some contamination of cells in that filtrate. And anyway, how could you catch cancer with such a tiny agent that you couldn't even see? And so he didn't get any leverage at all, and people were kind of very, very skeptical about his findings. He pursued it, however, a little bit longer, and it was really encouraged when he found another sarcoma virus in chickens, now called the Fujinami sarcoma virus, and that was also found in Japan by two researchers. And so then he was convinced that he was right, but he was so discouraged about people's not believing what he did that he decided to give up this whole area of research. We heard about that at TWIV, you know, when you can't get anywhere, just find something else to do. Well, that's what he did, and he had studies going on in liver physiology and immunology, so he was perfectly happy to do that. And then World War I came around, and he went in to help the war effort, and developed, in fact, the first blood-preserving methods. And this was very, very important, and I think that they use those same techniques today to preserve blood. It was extremely important in the establishment of the very first blood banks in the war that were used in the front line in Belgium. So he really did a quite important thing. But he went on with his physiology studies until 1934, when another virus that caused something that looked like a cancer was found in rabbits, the Shope papillomavirus. And his friend Shope brought him this wart, and he said, well, what is this? And being a pathologist, he knew, well, this is a benign cancer. And so he spent the next couple of years actually studying Shope papillomavirus, now convinced that viruses had some role in cancer. Well, you can see that Rouse did a lot of very important things in the war with the Shope papillomavirus. But what he's most known for, and what he finally got all of his prizes and the Nobel Prize in 1966, was actually for his work on the Rouse sarcoma virus, because that virus figured so importantly in the future years in cancer studies and actually molecular biology. So he got his Nobel Prize in 1966, 55 years after his publication. And it's been quoted as the longest incubation period for a Nobel Prize ever known. Well, in the years following that, in the 30s and 40s, retroviruses were found in many other animal species, mice and cats and so forth and so on. But although these cancers were found, it would be decades, really, until the properties of these viruses would be appreciated, their unique properties. And the deeper understanding, actually, would depend on research into the molecular basis of heredity, which was actually going on at the same time as these retroviruses were found. So, everybody today knows about DNA. It's on the news. If you watch television programs, you know that a drop of blood will be enough to convict somebody. And of course, you can get your own DNA sequenced and know about your ancestry. So it's really hard to imagine that it was not known until the middle of the last century how traits are inherited, and that traits or genes are encoded in DNA. And the progress was sometimes circuitous and took a very, very long time in what is called now the classical era of genetics. So, the founding father of this classical era is called, of course, Gregory Mendel. And he's also a very interesting fellow. He was born on a farm. He was a fairly poor fellow. He had two sisters, and he was pretty bright, but not very many prospects until, of course, he decided to enter the clergy. And he did that for a number of reasons, but mainly because he could get a very good education, and he could have a stable income the rest of his life. So it was a very practical decision. And so he got his education in physics and mathematics. So he was a physics person and a mathematics, but although, of course, living on a farm, he knew about plants and animals too, and he was always interested in heredity. So when he got finally to his abbey, he decided that he would like to do some experiments on heredity. And the first thing he wanted to study was mice. So he wanted to study inherited traits in mice, but the head of his abbey was not interested in having sex going on in animals in his abbey. So he decided, well, I think I'll do plants. So he decided to work with peas. And you can say, well, why peas? Well, the pea plant is very, very interesting because it's self-pollinating. And so you can get really nice, clear strains of various kinds of peas that are homozygous for whatever kind of trait you're interested in. And it had lots of traits, colors of the flowers and the seeds and so forth and so on. And over a period of about 8 years, he actually did about studies with about 30,000 pea plants. I mean, it was an enormous amount of work. And he was very, very careful in writing down all of his results. And being a mathematician and using very used to various mathematical models, he took all this data, which I guess it was big data at the time, and very different and very unique for botanists because it was a descriptive science at that time. He decided to kind of figure out what was going on. And as a result, he came up with three major observations, which are now known as Mendel's law. So, first of all, he found out that these individual traits, color of the seed or the texture of the seed, were inherited independently, like they were separate kind of entities. Then he found out that if he crossed two strains, homozygous strains, or with different colors, for example, one trait would dominate over the other. So he decided, well, one must be dominant, one of these traits must be dominant and the other recessive. And then he found that if he crossed the hybrids, these hybrids that formed had the dominant feature, if he crossed them, he could regenerate the recessive trait. And all of these things came out in particular ratios, which were very interesting. And they're actually summarized on this Punnett square here. So he found that if you crossed, this is not going. Oh, it doesn't shine on that. Okay, so if you cross these two hybrids with a dominant and a recessive and a dominant and recessive, the offspring, and of course he was looking at large numbers now, the offspring would distribute in a way that there was 3-to-1 had the dominant feature trait, and if you then crossed all of these hybrids again, you could discover that you could recover the recessive trait, and you could recover a pure dominant trait and two hybrids in a ratio of 1 to 2 to 1. So he's known, of course, as the father of genetics, but he was a very modest and differential man, and he wanted to publish his results. And so what did he do? He sent his paper to the most well-known, prominent botanist at the time, a guy called Carl Wilhelm Nageli, and he asked him to please submit it to this journal, not Nature, but the equivalent of Nature. And this botanist read this paper and couldn't make any sense out of it. He said it was completely empirical, irrational. He didn't believe any of it, and he told Mendel that he really ought to be looking at other plants. Well, the problem was that Nageli did not believe in Darwin evolution, and Mendel's results were really consistent with Darwin's evolution. So that was his first problem with it. The other problem was he was writing his own book on evolution, and this didn't fit with it. And third, he himself was doing experiments, genetic experiments, but he was using a plant whose seeds were almost all homozygous for one parent. And the reason for that was this particular plant produces a lot of seeds by parthenogenesis. So he had a completely different result, and he thought that Mendel's stuff was just kind of a fluke for peas. Well, Mendel never got his paper published in Nature, but he did present his results at a meeting, and the results from those meetings were actually published. And that's all we have from these very, very important experiments today. Now, a few other important things happened in that that first early period of classical genetics. Here we have Mendel with his experiments in 66. In 69, Friedrich Miesche identified a substance in the nucleus of cells, which he called nuclein, which is DNA. So this is the first discovery. This is the first discovery of DNA that we know of. And then in 1882, Walter Fleming, who was using a light microscope, but that was what they had in those days, was, come on in. So Walter Fleming was the first man to see chromosomes. And he saw these structures in cells that were doubling and moving during cell division, just as you might have predicted from Mendel's experiments. And then in 1900, 3 separate botanists, also working with pea plants and other plants, made the same discoveries that Mendel did. And they didn't know what to do about it, and then they found these old papers. And so Mendel was rediscovered in 1900, and really that sparked a new interest in genetics in general. Now, unfortunately, Mendel died 6 years before that. So he never got to know that people actually appreciated the work that he did. So now we're moving the pace up a little bit more. In 1902 and 1903, 2 scientists, Walter Sutton, working with insects, and Theodore Boveri with sea urchins, noticed that Sutton noticed that chromosomes would separate they would line up in pairs and then separate to each cell as cells divided, just as again would be predicted from Mendel. And Theodore Boveri noticed in sea urchin that unless the embryos had the right number of chromosomes, they failed to develop. So this again was consistent with the fact that genetic material or heredity existed in chromosomes. And then in 1910, T.H. Morgan showed that eye color in Drosophila is sex-linked on a chromosome. And he also developed linkage maps. So he showed that the traits were linked one to another. Well, the idea that chromosomes have genetic material was clinched right here at Cold Spring Harbor when Harriet Crichton and Barbara McClintock showed with their corn studies that there is a relationship between genetic breakage and recombination of genetic results. So chromosomes have the DNA, have genetic material. That was clear. But chromosomes are made of proteins and DNA. And everybody presumed at this time that traits or genes were made of proteins. Now, why should that be? Well, it is pretty logical. Proteins contain 20 amino acids, and you can recombine them in all different ways, so you can make an infinite number of combinations. And people also knew that proteins had biological activity. They had enzymes. So obviously, now DNA was totally boring. It had only 4 bases, and it was these long chains of nothing. So the idea that was cemented in everybody's mind at the time was that genes are proteins, and the DNA is just a scaffold. Now, that idea was clashed when Avery, McLeod, and McCarthy showed that they could transform pneumococcal bacteria with DNA. So this Avery, McCarthy, and McLeod experiment really marks the end of the classical age and the beginning of molecular biology. So what was Avery's experiment? He was fascinated by an earlier report in 1928 that you could mix extracts of killed pathogenic pneumococci with pneumococcus that was not pathogenic, and you could induce the non-pathogenic ones to become pathogenic. And that was fascinating, but that observation just sat there. So Avery decided to find out what that factor was. And he was going to do it by a process of elimination, because by that time, we had enzymes that could attack different components of a cell. So his idea was to make the extract, to challenge it with lipases, with proteases that would kill the protein, and then with DNases. And when he used the lipase, he got nothing. The transforming principle was still there. When he used the protease, nothing. But the DNase killed the transforming activity. So then he thought, well, what if I use pure DNA? And he lysed the bacteria and he purified the DNA. And he did this experiment that's shown here. So here are the non-pathogenic bacteria, and they make these kinds of colonies. If he took DNA from these pathogenic strains and mixed it with the bacteria, plated them out, he got pathogenic colonies. And he could see them because they were large and smooth. But if we treated the DNA with DNase, he got no pathogenic colonies. So he said, he printed this, he published a paper, and he said, genes are made of DNA. Well, he again was totally ignored because everybody knew that genes were made of proteins. So this person must have been mad. And he wasn't helped by the fact that a colleague of his, Mirsky, who was actually also in Rockefeller, published a paper the following year which claimed that even 1 to 2% protein contamination of his DNA would have given him the same results. So there he was, and he continued to do experiments, but was generally ignored by the field. So then comes along Alfred Hershey. So I can tell you that Alfred Hershey was also a person who believed that genes were made of chromosomes. But he was working with bacteriophages, which are very simple. They're made up only of protein and DNA. So he decided, well, maybe I can figure this out or prove it using this system. And he was eager to do that because at the time, isotopes were just becoming available that would specifically label proteins or DNA. So he used S35 to label the protein of the phage. He grew up the phage in S35 containing medium. And he used P32 to label the DNA. And then he put each of these, this S35 and P32 DNA, he infected cells with them, and quickly, after allowing attachment of the virus, he put them into a wearing blender. And I put this picture here because this is the actual blender that he used, and we have this in this library, actually. So you can take a look and find it if you wish. He put it in the blender, spun them, and then he separated the bacteria and the supernatant. And what he found was most of the S35 was in the supernatant, and most of the P32 was in the bacteria. Wow, that was not what they expected. But it was even worse than that. When he looked at the phage that came out, the progeny phage, these phage were labeled with P32. There was no S35 here. But if you look at the numbers, you know, they're not great. They're actually worse than Avery's numbers. So if you look at S35, well, most of it is in the supernatant for sure, but there's a lot in the pellet. Some of that was just adventitiously held there. And if you look at the P32, well, most of it is in the bacteria, but there's some in the supernatant. The clincher, of course, is this one, that the progeny phage contained 30% of the input P32. But it was enough to convince the world, which is kind of surprising. And not Hershey, though. He was not convinced. And I put this here. This is the last sentence in his paper. And what he said is the sulfur-containing protein, and he was careful to say that because he figured, well, there may be some protein that doesn't have much sulfur. Well, the sulfur-containing protein is confined to a protective coat and probably has no function in the growth of intracellular phage, right? But the DNA has some function. Further chemical inferences should not be drawn from these experiments, right? He was convinced that proteins still had some role in there. And he was talking to Alan Campbell shortly after this experiment and its acceptance generally. And what he said was, I predict that DNA will not turn out to be the only genetic substance, and also that the most important progress in the next few years will be made by those willing to assume that it is. So he was not, even at the end, not convinced. And he was asked a little bit later by Václav Jabalski, this guy here, why he didn't believe, why didn't he believe Avery? Because nobody in America, he said, we believed it in Europe, we believed Avery's. And the response from Hershey was, nobody in America believed their data. Mirsky convinced everyone that Avery et al. Did not know the difference between DNA and nucleoprotein. So it's really really interesting. If you go back to Hershey's paper, this famous 1952 paper, he never mentions Avery. There is no reference to Avery, which is really was a surprise to me when I went back to look at it. So the progress was very, very rapid by those who believed that DNA was the stuff. And so in the next 10 years, we had, decade, we had enormous progress. First, Chargaff discovers that A equals T and G equals C, and then Watson and Crick use this information and Rosalind's, Franklin's beautiful EM picture to make a structure of the double helix. And George Pilati sees the ribosomes in cells, which was subsequently shown to be cytoplasmic protein factories. Then Arthur Kornberg discovers the first DNA polymerase from E. coli, Pol1. Messels and Stoll show that replication is semi-conservative. And Brenner, Crick, Jacob, and Monroe predict the existence of mRNA, because if DNA is in the nucleus, how does the information get out to the cytoplasm, to these factories, right? And there was a discovery the year before of something called adapter DNA, tRNA, that could make the difference, right? Finally, in 1961, Nierenberg et al. Cracked the genetic code. 1 decade, it really went fast. So that was the beginning, that was called the golden age of molecular biology. And so a fundamental principle of life on Earth was established. DNA makes RNA, messenger RNA, and that goes into the cytoplasm and is translated in ribosomes to protein, right? Unidirectional flow of information. So that pathway became universally accepted as the central dogma. Everybody bought in. It was obvious, right? And when you think about it, it's really impressive. So here we have the whole Rosetta Stone of genetics, that the whole code, and you can just put it on one piece of paper like this, and all life on Earth uses the same code. Fantastic, right? I mean, this is the basis of how we can do all of our evolutionary comparisons now with this big data, right? And this, of course, supported the idea that was first proposed by Darwin, that all branches of the tree of life descend from an original ancestor, right? So, all important principles are established. All we have to do now is fill in the blanks. Okay, little did anyone know that this dogma was going to be amended. So the first wrinkle came in the mid-1950s, because it was found out that some viruses don't contain DNA, they contain RNA. Actually, they're the only biological entities that have RNA in them. So they don't have any DNA. So what's going on here? Maybe for these viruses, it is protein as the genes, right? So Heinz Frankel Conrad and his colleagues did a fantastic experiment with TMV. It's amazing when you think about it. It's a simple virus, and you can isolate the protein and have that in one flask, and the RNA in the other flask, and nothing happens. But if you put them together and wait overnight, you get particles that look like the original particle. They simultaneously form. Not only that, but the particles are infectious. So they thought, wow, this is a way to find out who's who, right? So they had different strains of TMV. They could distinguish them because their proteins were slightly different. And they decided to ask, well, what is the genetic component here? So they mixed and matched the RNAs and the proteins from these various strains, got particles, and then asked. And what they found was the protein that was found in the progeny depended on what RNA you used. So for these viruses, RNA is the genetic material. Well, that's not so bad because it doesn't go against the directionality, right? Viruses are just a little different. They don't bother with DNA. They just go for it directly from RNA to protein. So that was okay. But then a young man studying in Wisconsin, or studying in actually Caltech, he was studying another virus, the Rous sarcoma virus. So that's the one that we talked about earlier. And that he found that different strains can make distinct genetic changes in cells, which are transmitted to all daughter cells as they divide. Now, how can that be if cellular genes are DNA? Because these are RNA viruses. So you infect with this RNA virus, and then you get a transformation, but that's constant through all the generations of these cells. So Temin hypothesized that either that the RNA genome of the infecting virus somehow acts as a mutagen for the DNA, or it is somehow changed into DNA, which he called the provirus. So he proposed further that the provirus persists as an integral component of the genetic inheritance of its host cell, where it provides a template for the synthesis of progeny virus. So this was not a new idea, because we knew from phage work that there are some phages that can integrate DNA into the cells, and then they become a genetic component. But in that case, they don't continue to do progeny. They come out again if they want to make new phage. So it's slightly different. Anyway, for most of his contemporaries, this was a wacky idea. And there couldn't be any exception to this unidirectional flow. And so he was dismissed by most people. He was scorned by some. But essentially, he was disregarded for 6 years, even though he continued to do his work. Now, it turns out that Howard Temin wasn't alone, because there was another scientist working in Prague, which then part of the Soviet Union, Jan Svoboda, who was also making similar observations, right? So he had discovered that there was a strain of Rous sarcoma virus that could transform rat cells. And these rat cells then would be dividing, and they would never produce virus. But if you injected them into chickens, you would get virus back. And the virus would be the same as the one that he had used initially. So he called these cells virogenic, in analogy with lysogenic, right? And so the two of them exchanged correspondence, and they were trying to do experiments to prove that what they thought was correct. But the technology for the day was not really up to it, and they really could not convince anybody until the late 60s. a number of other virologists had discovered that virus particles sometimes include enzymes in their particles, right? And one of them was a DNA virus, a pox virus, included a DNA-dependent DNA polymerase. And another was an RNA virus, reovirus. It included an RNA-dependent RNA polymerase. And Baltimore decided to try another RNA virus, VSV, that was being worked on by his wife, Alice Wang, and the two of them discovered that this VSV also has an RNA-dependent RNA polymerase. And so the two of them were very interested in this fact that you could find enzymes in particles, and they decided to look in retrovirus particles. Baltimore separately, he got a batch of RNA viruses from NIH, and of course Temin was routinely working with RSV in his laboratory, and he had a biochemist postdoc, Shitoshi Mitsutani, who was eager to try. And they independently found that indeed retroviruses have an DNA-dependent RNA polymerase, which was named as RT, reverse transcriptase, by Nature when their paper was published. So this is the amending of the dogma. You have RNA that can go to DNA, and then it goes through the regular cycle. Well, the unique technical capabilities of these retroviruses also brought new questions into focus. Where did this ability to bridge two genomic worlds, the RNA genomic world and the DNA world, come from?
Can I ask you something?
Yes.
Why was that so quickly accepted?
Oh.
I never understood how something as crazy as reverse transcription could go from publication to Nobel Prize in 4 years.
4 years.
Yeah.
Yes. 55 years for poor Rouse and 4 years here. It was simultaneously independent. It was uncontroversial. I mean, it was clear. There was this enzyme that did this thing. There was no question about it.
And like you said, by this time it was clear that many viruses contain enzymes. And
Yeah.
we accepted that negative strand viruses.
Yeah.
So,
Yeah. Yeah.
It wasn't only.
Okay, so origin of retroviruses. The question of how life began on Earth has mystified people throughout the ages, and numerous myths and diverse religions have offered explanations not only of who was involved, but how it transpired. The question of who is a question for theology, but science has managed to make some inroads into the question of how it transpired. There is general agreement that RNA played a major role at some very early time in the evolution of life, as we now know it. In this RNA world, as it is called, primitive RNA chains are thought to have arisen spontaneously with support and energy from some promoting feature of the environment of primordial Earth, like deep sea hydrothermal vents or tidal pools. And these RNA chains could multiply by serving as templates for bases that could pair with available nucleic acid substrates. And successive rounds of this mode of replication would therefore be the basis of genetic continuity in these chains. So one can think of them as selfish mini-genes whose function in their own reproduction. Now, occasional mistakes in base pairing would then be responsible for driving their evolution. The most stable RNA chains and the most efficient at making copies would gradually outnumber all the rest. Now, it's also conceivable that small molecules, such as peptides or various metals and other inorganic substances in the environment, may have supported such reactions. But in this world, RNA served as both the repository of information, genes, and catalyst enzymes. So, RNA can do it, and RNA genomes are available. Why do we have DNA? Why didn't this persist? Well, there are a couple of reasons. DNA is 100 times more stable than RNA. Just the substitution of ribose for deoxyribose for ribose makes the chain 100 times more stable. So that oxygen, just one oxygen removal. That means you can make longer chains and get more information in a single strand. And also, DNA makes double strands. So if there is a mistake in one strand, it can be corrected by copying the complement, right? So we don't know how or when the transition from RNA to DNA world occurred. Some speculate that it happened even before cells or viruses existed in the primordial soup. But the discovery of RT provided a glimpse of how this transition may have been facilitated at some time. Now we know that both RT and integrase, the other enzyme in retroviruses that is so important, are very ancient enzymes. They're present in viruses and transposable elements in all branches of life. And it came as a shock to everyone when the first human genome was studied, that the protein encoding sequences containing the information for all of the proteins in our bodies account for only about 1% of our DNA. But in mind-boggling contrast, half of our DNA was found to be made of transposable elements or repetitive sequences derived from them. So DNA transposons, as we've shown here, comprise 3% of human DNA, while a whopping 42% of our DNA consists of retrotransposons. And they use RT to make copies of DNA, they inserted DNA, and then integrase or transposase to insert these new copies at other locations. And 8% of our DNA are ancient retroviral sequences that have been accumulating in the genomes of our evolutionary ancestors over millions of years. In fact, you're absolutely, absolutely. So in fact, recent computational analyses indicate that approximately half of the remaining portion of our genome, the dark matter, is also composed of previously unrecognized repetitive sequences derived from transposable elements. So while there are lots of mysteries to be explored in this fantastic landscape of our genome, it's hard to escape the humbling conclusion that most of our genome is nothing more than a collection of transposable elements and byproducts that have accumulated over evolution. And we're not alone. An astounding 50 to 90% of the genomes of many animals and plants are made up of such elements. Okay, so we're getting late on time, so I'm just going to show one more slide, and that is an idea of getting the perspective that we're just talking about of all these events in evolution. So the latest estimates are that the RNA world could have started 4.4 billion years ago here, shortly after Earth is formed. And the earliest evidence for microbial life is about 3.4 billion years ago. So we have 1 billion years to go from the RNA world to DNA, right? Now, all animal species are host for retroviruses, so most must have arisen early in evolution. And the earliest known retroviruses are known to exist on animals that were present here during the Paleozoic era. And we can show that here, 542 million years ago. So these are animals that are in the ocean, and they haven't arrived yet on land. Now, that means that, of course, the retroviruses must have been around before that. Okay, so I'm going to end here. There are several more chapters we got here, but I'll leave this to the rest of the people who might like to read the book. It's full of these kinds of stories, and I'll give you the last chapter, which is Epilogue, which I try to pull it all together, but I talk about progress in treatment of retroviral diseases, retroviral vectors, the use in directing evolution by making transgenic plants and animals, and I have these final reflections. And I'm open to any questions that you might have.
At what chapter did you enter? Did you start reading?
I ended at 3, I believe.
At the end of 3 or beginning of 3?
The next one would have been retroviruses and evolution. So what I've talked about, endogenous retroviruses and what they do, and I would have talked about their role in placenta formation. Without a retrovirus, we might be laying eggs like birds. And this is a genetic basis of cancer. This is using Rouse sarcoma virus again, finding Sarc and oncogenes, finding the MYC oncogene and insertional mutagenesis, and of course the first human retrovirus, HTLV1, and what all that did to help us in this last episode, which was HIV, where it came from, what it does, what we can do about it, all the controversies and so forth. So that's the rest of the book.
I think Viviana was asking when you came in, between chapters 1 and 2, right?
When I came into, I was here with Hershey. I was here with Hershey from 64 to 69. So it was after Hershey and Chase, but he still was convinced that proteins were going to be important. And he's right. Proteins are important in heredity, aren't they? We know now, right?
So is there an increase in the percentage of the genome of species as you go up the ladder, or not?
No.
We were all bombarded with cancer viruses from the beginning.
Right. I mean, but it's just extraordinary when you think about it. It's scary.
The biggest genomes are in plants, right? Or at least some.
Plants, 90%.
Yeah.
Yes.
There is like a
retrotransposons. Yeah. Okay, let's have some wine and cheese.